I will be the next speaker, so I will present myself. I'm Natalia Freund from the Department of Clinical Microbiology and Immunology in the Faculty of Medicine of Tel Aviv University. And I took the liberty to change the title of my talk compared to what you have in the flyer, so I will give an overview on our work on B-cell and antibody responses to pathogens, and towards the end I will talk about our recent work on monkeypox infection. Okay, so this is our galaxy, the Milky Way, and it has an enormous number of stars, 100 billion stars in it. And this number is actually 10,000 times smaller than the number of different B cells that we have in our bodies. So we have an enormous diversity in our B cell repertoire. And when I say different B cells, I mean that the B cells express different antibodies on their surface. So according to Frank Burnett, who was an immunologist and defined the clonal theory in the middle of the previous century, an antibody that is expressed on the B cell defines the nature of the B cell. So we have a huge diversity of different B cells, meaning that B cells can express different antibodies, enabling us to bind different antigens and to cope with different threats that we will encounter throughout our bodies, throughout our lives. So this naive repertoire is extremely potent and diverse. However, it can be viewed as a collection of babies who hold a lot of potential, but in fact, when it comes to defending us from threats, they are pretty useless. So in order to become fully active, they need to be stimulated. And this stimulation occurs by exposure to an antigen in a form of infection or a vaccine. And once a B cell recognizes through its membrane antibody an antigen that belongs to a pathogen, it becomes stimulated, and then it goes and generates these extremely fascinating structures that are called germinal centers, which are in fact like universities for B cells. This is where B cells mature, they mutate, they improve. Eventually, they're being tested for their membrane antibodies, and only the B cells that express high affinity antibodies on their surface, which are also very stable and active, they become selected, they survive, and eventually they become these antibody secreting cells, the plasma cells that we can see in the serum a few days after the infection. But the truth is that plasma cells do not stay for very long times in the bloodstream, but rather they go elsewhere, they disappear, and the cells that remain are these avengers, the memory B cells that stay as long as we live throughout our entire lifetime. And these might look like the naive B cells that they originate from, but in fact they are very different. They are much more active. They contain high affinity antibodies. They come in families, in clones of B cells, and they are the ones that will be able to protect us during secondary encounter. So my lab is interested in these memory B cells, and we ask questions how memory B cells can be linked to different diseases and how different diseases shape our memory B cell repertoire. And we ask many different questions. For example, do antibodies work in teams? Or are they single players? Do we have a specific memory B cell type or specific antibody type that addresses a specific disease? Or rather, do we have different types of antibodies that cooperate with each other? Do we have different types in different people, or do we have convergence in the same response to the same disease? We ask how much affinity maturation is important. Do all diseases need this kind of education, or do we have some of the repertoire within our naive B cells? And does strong antibody response necessarily correlate with reduced disease or not? So we're trying to answer these questions, and we will talk about them today during the talk by looking at 4 different disease models. So we'll start with HIV, then we go to SARS-CoV-2, we'll talk about Mycobacterium tuberculosis, and we will finish today's seminar with the monkeypox virus. So in my lab, we use the single-cell sorting technique, which is a fairly simple approach that we call the learning from the body approach. So why is it learning from the body? We don't work with engineered antibodies. We look at the antibodies that are naturally produced by patients. We collect blood samples, and then we single-cell sort them using this very cool sorting device that puts one cell into one well. How do we collect? Okay, nope. Back. How do we collect these cells? So this is. Okay, so we basically stain the B cells with disease-related antigens, and this is how we isolate only the memory B cells that we are interested in. And then every memory B cell that is related to the disease we want to identify, we want to study, goes into one well. We then amplify the antibody segments by PCR, by single-cell PCR. We can just then sequence the antibodies, we clone them into expression vectors, we produce them, and then at the end of this process, we have naturally produced antibodies. Then we can then assess in various assays, try them in mice, in vitro, use them as candidates for drugs, for vaccines, or for diagnosis. Okay, so let's start talking about our model systems for infections today, which are also very relevant to pandemics, because the first pathogen I want to address is human immunodeficiency virus, HIV. And HIV is an unsolved problem by science. Why? Because there is no cure and there is no vaccine. And why is that? So first of all, very briefly, HIV has enormous mutation rate, the highest recorded in nature. One base in every 1,000 bases is a mistake. And I'm showing here the global variation of influenza A virus, and on top of that, the variation in just one patient, who is HIV infected, and you can appreciate that it is very high. The second reason for HIV being challenging is the fact that the only protein that is expressed on the surface of the virus, the envelope protein, is covered by these green residues here, which are glycoresidues, and the virus uses glycans similarly to the way Harry Potter uses the invisibility cloak, so it becomes invisible to the immune system. The third reason is that the HIV virion expresses a very small number of these env proteins. So the virion is almost bald, expressing 5 up to 25 functional envelope proteins per spike, and this makes it very difficult for antibodies to see this virus, and of course averts any possibilities for avidity. Nevertheless, 5% of HIV-infected individuals develop these neutralizing antibodies that can get the virus. And we heard from Tzachi about viral evasion. So some antibodies are able to overcome this evasion. And these are called elite neutralizers. And I will show them in these heat maps so that I will take a minute to explain. So we test a panel of viruses, in this case, 7 different viruses. And our assay is inhibition of infection. So we test how potent the antibodies in the blood of the donor are to inhibit 50% of the virus. This is IC50. So red color indicates high neutralization, yellow low neutralization, and white would indicate no activity at all. So we asked what antibodies contribute to viral control. And in this work that was done still during my postdoc work, we analyzed the lead controller patient, EB354. You can see the viral load of this patient. It spikes a little bit, but since 2006, it becomes clinically under detection or very low viral load. During this time, the donor develops neutralizing response. So here you can see we have a new color, cyan, which means exceptionally high potency of the antibodies, and the antibodies become more and more neutralizing as we progress through time. During this time, we were able to isolate 3 neutralizing antibodies named NC37, BG18, and BG1, and you can see their neutralization profiles here. So each antibody has a different neutralization profile, but together they managed to recapitulate the neutralization profile of the serum. The antibodies were extremely mutated. So when we look on the sequences of the antibodies and compare them to the naive B-cell sequences, we find that about 50% of the antibody is changed. That indicates that the antibodies need to undergo a very complex evolutionary pathway to reach this kind of activity. So this is a 180 viral panel. You can see here many different strains of HIV. Every line is a virus. And you can appreciate that these 3 antibodies are extremely potent. The most potent antibody is this one on the right side, BG18. And together, these antibodies reach 81% of coverage of all known HIV strains. So they have different neutralization profiles, and this already was telling us that the epitopes that they're binding are different, and we did a lot of structural work, and here you can see it summarized in one cartoon or one model. This is a cryo-EM picture of the envelope from the side view, and these are the 3 antibodies, so they all bind in 3 different sites, and this was validated by mutations that you can see here on the bottom. So NC37 is a CD4 binding site blocking antibody, BG1 binds from the top of the trimer, and BG18 binds to the V3 loop glycan. These antibodies are active, but can they really suppress HIV? So for that, we use the mouse model. As you know, HIV does not infect mice. So in order to carry out mouse experiments, we used humanized mice. We used RAG knockout mice that were irradiated and reconstituted with human hematopoietic stem cells to regenerate a human immune response in mice. These mice now can be infected with HIV to generate stable viral loads that are very similar to the ones we see in humans. You can see here, 3 weeks after infection, we get baseline of HIV levels, around 10 in the level of 4 to 10 in the level of 6 viral loads in these mice. Now we can infect these mice with HIV and treat them with our antibodies. So I'm going to show one antibody, BG18, which is also the most potent one. Here, all viral loads were normalized to 0, and we are going to see the reduction in viral load following antibody treatment. So all the filled symbols are treated mice, the empty symbols are controls, and the red full and the dashed line indicate averages of the experimental and control groups, respectively. So you can see here, so once we start treating, the viral load drops down, as we would expect from a neutralizing antibody, but after a few, 2 weeks, the viral load goes back up, and that indicates an escape. When we sequence the viruses from the treated mice, we can indeed see that all the viruses compare point mutations in the epitope of the antibody, and this, of course, indicates that the viruses escape the antibody. What happens when we treat with all 3 antibodies? So I don't show it, but the other 2 antibodies generate exactly the same result. So when we treat with a combination of the antibodies, we can see that viral load goes down, and we do not see any escapes. So viral load stays down, the mice do not rebound. Okay, and this is statistically significant, and even after we stop treating, we still see that viral suppression remains. So these antibodies are very efficient, and at the end of this work, they were licensed along with other antibodies in the lab to Gilead Biosciences that are now doing clinical trials in humans. Okay, so HIV, let's see how common the antibodies against the pathogen. So not common, okay? We saw only 5% of people can even produce these antibodies. They work as a team, so we need different antibodies tackling different sites simultaneously in order to get the effect. They require extensive affinity maturation. They do correlate with disease control because we saw the patient is suppressed, and their mechanism of neutralization is blocking the infection. Okay, so now let's go to the next model, and this is, of course, SARS-CoV-2. And here, this work is led by Michael in the lab, who is a PhD student graduating this year already. And together we looked at the antibody responses in severe versus mild COVID patients. And we asked whether severe COVID correlates with better antibody responses or worse. So here you can see the antibodies collected from the donors that were analyzed in this work. And we saw that maybe the opposite of what we initially thought, mild disease does not correlate with better antibodies, but the other way around. So more severe disease correlates with higher antibody responses, and not only in the serum. So we use 10x platform to sequence B cells from these donors, and we could see that severely infected donors have more clonal expansion, they have more shared clones, so they have more significant, robust antibody responses. We also identified a genetic feature that is common to severely infected patients and to response to SARS-CoV-2, and this is VH3-53 that was identified in this work and also confirmed by other labs. Lastly, we isolated 9 neutralizing antibodies from 2 severely infected patients, and these antibodies were then characterized. So the antibodies could be divided into two different groups according to their mechanism of action. One group blocked the receptor binding, and these were the ACE2 blocking mAbs, and the other group neutralized but did not block the receptor binding. So it neutralized the binding. It prevented the entry of the virus, but not by preventing attachment. So as the variants came along, we started assessing these antibodies against the new variants. And you can see here something that was very typical. So the wild-type virus, all the antibodies were active against it. It looked wonderful, and I know a lot of monoclonal antibodies were cloned around this time. Some of them progressed to the clinic. However, when the variants came around, some of these antibodies became inactive. And you can see it here. While we lose only a little bit of activity against the alpha, when we look at beta variant, gamma, and delta, most of the ACE2 binding site antibodies become inactive. However, 3 out of 4 non-ACE2 binding site antibodies still bound and neutralized. So this is data about the Omicron variants, and you can see here that including the recent XBB variant, we get a pretty nice coverage of our neutralizing antibodies that are non-ACE2 binding site. And I want to note here that while there were thousands of antibodies cloned from the beginning of the pandemic, there is only a handful of antibodies that are active against all the variants. And for this, we are extremely excited to have some of these in our lab. So we did some epitope mapping, and this was done by phage display and AlphaFold. And we think that the antibodies that are conserved, they bind to a site which is in the side of the RBD, not where the receptor binds ACE2. So we're still elucidating the structural characterization. Okay, so let's go back to our table. So SARS-CoV-2, the antibodies are very common. The majority of people who are infected can produce them. They work in team, so we see that there is a contribution to different mechanisms of neutralization. I didn't go through it, but the antibodies are not mutated at all. So we have only 1 to 5, sometimes 10 mutations per sequence, so they do not require a complex developmental path like HIV antibodies. They do not correlate with disease control, the opposite. So people with more severe disease get higher stimulation and produce these antibodies better than people with asymptomatic infection. And the mechanism would be, again, blocking the infection, similarly to HIV. Okay, I want to switch gears to talk about complex pathogens. And when I say complex, I mean pathogens that are not as small and expressing only one protein on their surface. So both HIV and SARS-CoV-2 are relatively fit for antibodies to work because antibodies can cover them relatively easily. And also, it's pretty clear what the target for the antibodies, what the target is. However, when we talk about larger viruses, such as the smallpox and pox viruses in general, these are way larger and express many different proteins on their targets, so it's not clear what antibodies should bind in order to neutralize these viruses. This becomes even more complicated when we talk about bacteria. There are whole organisms producing sometimes thousands of different proteins. What antibodies should bind to neutralize them? We don't know. Not only they express a large abundance of proteins, these more complex pathogens most of the times do not enter the cell by receptor-mediated entry, so there isn't one key that goes into one lock to open the door into the cell, but rather a collection of different antigens, and we don't know which ones are the more important. For bacteria especially, some bacteria reside inside the cells, while antibodies are outside. So for these reasons and others, we kind of became interested in more complex pathogens, asking, do antibodies even contribute to control of complex pathogens? And I'm going to start talking about TB, and I will be very brief about that because Avia, our PhD student who is sitting here, she will be giving her student presentation today at 4, so you're all very much invited to stick around and listen to this beautiful work. So Avia is working on TB in my lab, and also Lilach Abramovich, who is a staff scientist in the lab. And here I think that we managed to make some real impact in the field, because before we started working on that, it was pretty much taboo that in Mycobacterium tuberculosis, it's only T cells who contribute to bacterial control. And some literature that came out during the last years, including from our lab, proved that antibodies may also have some role, and I will try to tie it a bit together in the next few slides. So Lilach and Avia identified, using protein arrays, PSTS1 as one of the targets for antibodies in Mycobacterium tuberculosis. So you can see here a cohort of Israeli recruited patients. Every patient responds to different pathogens. These are the bacteria, these are the cloned antigens, and PSTS1 was identified as one of the targets for antibodies. So PSTS1 is a phosphate pump that is located on the inner surface of the bacteria. Using single-cell sorting, we cloned anti-PSTS1 monoclonal antibodies, and we had a bunch of them, but I will focus on 2 antibodies. So the antibodies bind PSTS1 with high affinity. They bind pathogenic TB. These antibodies were extremely mutated. So they were not as mutated as the HIV antibodies, but they contained quite a lot of mutations in their sequences. And when we asked, do they need these mutations, we found out that indeed these mutations are needed for the antibody activity. And you can see it here. So the red, the field marks, the field symbols indicate the mature antibody, and the open circles indicate the antibody where we reverted all the affinity matured mutations. We basically used the naive antibody. And you can see that in the case of this red antibody, the activity is greatly reduced when we cancel the mutations. And in the case of the blue antibody, there is complete knockdown of the activity. So it needs affinity maturation for activity. These antibodies inhibit pathogenic TB. These experiments were done with a colleague of ours in UCSF. They inhibit TB in vitro. This is an in vitro assay, Mycobacterium growth inhibition assay. The inhibition is not 100%. It doesn't look the same as for the HIV SARS-CoV-2 inhibition assays. So we can get about half of the pathogen knocked down. It is significant, but it's not 100%. Similar data is seen when we do the in vivo experiments. So when we give the antibodies prophylactically, infect the mice with the pathogenic bacteria, sack the mice, take out the lungs, we can see a reduction in the bacterial loads, but again, we cannot reach 100% here. I want to point your attention to the yellow bar here. So we combine the 2 antibodies together. We still do not see an additive or synergistic effect. So this is as good as we can get, probably. The mechanism of neutralization here is not entry inhibition, but rather there is an enhancement of entry. So antibody-mediated cellular phagocytosis, the antibodies take the bacteria into the cells. When we knock down the FC activity, we can see inhibition of the antibody activity. So we block the receptor by antibodies, or we blocked the antibody FC region by mutating it and preventing it from binding to the FC receptor, and we can see that the antibodies no longer inhibit. What the antibodies are doing inside the cells, okay? So they're taking the bacteria into the cell. What are they doing there? So we have some evidence that the inhibition is caspase-1 dependent. It is dependent upon activation of the inflammasome in the cells. And we also know that in mice, antibodies are recruiting T cells. So we know that T cells are super important, and we know that antibodies can recruit immune cells, and in the case of TB, they are recruiting TB-specific T cells, which can sort of clarify why both T cells and B cells are very important. Okay, summarizing the TB story, the antibodies are not common. If you remember the protein array, only some 10% of the patients can make these anti-PSTS1 antibodies. We still don't know if they work in TB because we only found one target. We know that TB has many proteins, so probably other antibodies against other proteins can contribute to protection as well. They require a lot of affinity maturation, as we saw from the mutations that they have. They do not correlate with disease control, so the patient from which we isolated these antibodies was very, very sick. So again, that indicates that to make antibodies, you sometimes need a lot of disease load, a lot of pathogenic load. And they do not block infection, but rather they engage FC-mediated functions. Okay, our last player for today is monkeypox, and this project is led by Ron in my lab, an MD PhD student. And this work was started a year ago during the outbreak of the monkeypox infection. We recruited 4 groups of donors. This was a collaboration with the Ichilov Hospital and highly supported by Tel Aviv Vice President for Research and Eli Sprecher, who is the VP R&D of Ichilov. And we ask whether recoveries, recent vaccinees, past vaccinees have similar immune responses. And you can see here binding assay for the cousin of monkeypox, who is the vaccinia virus. And you can see that recoveries bind very strongly, but also vaccinees bind. And interestingly, people who were vaccinated 5 decades ago against the smallpox virus still have high titers of antibodies. What about neutralization? So this is again the same heat map that we saw a few slides ago in the HIV project. So colors mean good neutralization, inhibition of the virus. And we can see that we get quite a lot of neutralization here, but only if we add complement. So without complement, we get some activity, but this activity is greatly enhanced when you add complement factors to the neutralization assay. We were interested in targets for monkeypox antibodies, and Ron cloned a lot of proteins. Here I'm showing you 3 proteins, A35R, M1R, and H3L. All of these are from the monkeypox genome. We produced them and tagged them, and we could see that for some antibodies, for 2 antigens, H3L and A35R, we get a much stronger response amongst the recoveries compared to the vaccinees. And this is not only in the serum, so we only get a lot of specific B cells. And now our next step is to purify antibodies. I'm not going to show that. Yeah, is to purify antibodies by single cell sorting. And this is very much preliminary because we now have we're now in the process of characterizing these antibodies and producing them and testing them. So hopefully we will have this data soon. Okay, so monkeypox. Antibodies look pretty common, so recoveries produce antibodies. They're not rare. We still, again, don't know if they work in team or single players. This is in the case of complex pathogens where we don't know all the targets. It doesn't look like they're highly mutated, so they are pretty much close to germline. They do not correlate with disease control, so again, we can see severe infection and people producing these antibodies still. And the mechanism is probably complement dependent. So it's not a simple blocking of infection, but it requires the immune response. Okay, take-home messages. So antibodies against pathogens work in different pathways. It's very much custom tailored to the disease you're studying. The antibodies will behave differently. Some antibodies require more developmental pathways than others. For example, HIV needs a lot of mutations, so does TB. On the other hand, SARS-CoV-2 and monkeypox do not. Antibodies can block infection simply by binding, or they can engage the immune response by their FC portion. Both are important. And good antibodies do not necessarily indicate a mild disease. So if you're very sick, it might indicate that you're producing very good antibody response that will protect you next time you encounter the pathogen. So it's not necessarily that a sick person cannot be a source of very good antibodies. Our current direction in the lab, so our TB project, we work on improving the antibodies, we work on engineering of these antibodies. Lilach is leading our immunization with PSTS1 project, which is also supported by the Gates Foundation, and we collaborate with BioNTech on that. We have new targets for SARS-CoV-2, which is the work of Michael. We work still on monkeypox. We have 2 more projects in the lab that I did not have time to go into. Okay, so I would like to thank my amazing team, who is really a collection of really smart, brilliant, motivated, and fun people. And it's really fun to come to the lab every morning and see people who are excited about science and life also. I want to thank our collaborators and funders, the foundations, and we have a lot of collaborations with the industry. And of course, a very large group of collaborators. So we are a B-cell lab, but we collaborate with structural biologists, with virologists, clinicians, microbiologists, computational biologists. And all of these people are really contributing and contributed to this project. So thank you very much, and I will be happy to take questions. Tzachi. So you're asking whether NHPs, if non-human primates, produce similar antibodies.
Or different, because some of them are known to...
Yes. So it's a bit hard to study HIV in... so NHPs are the closest to humans, and they are used a lot to study for therapy, trials. But to study the evolution of HIV in human primates is a bit challenging because, as you said, the disease is very different. So they do not develop the same kind of immunodeficiency as humans. It's much milder in macaques. We know that they produce antibodies, and there is a lot of research in that. So they do produce antibodies. They are usually not as mutated, so they are more easily produced than in humans. So they do get neutralizing antibodies, but still, again, they cannot clear the infection because of the latent reservoir. Yes, Cyril. So I think it's a very exciting finding that antibodies recruit T cells, because it can settle down the argument of B cells or T cells, and the answer would be both B cells and T cells. So we know that neutrophils are also very important for TB, and we see a lot of resemblance between granulomas in TB and cancers if we look on cold versus hot. So the more immune infiltration you get, it's better for the disease course. You can destroy TB better. I cannot say it. Yeah. Yes.
Do you know which type of T cells are coded to kill the pathogen?
In TB?
Yeah.
Yes, this was just asked. I don't know. Yeah, I don't know yet. Yeah. Okay, thank you very much. Thank you to all the speakers today.